y'all for being here, and thank you for leaving the poster session. I know that was an excellent time and lots of good conversations, and hopefully I can keep up the energy with that and have some fun conversations with this. This is a topic that we like a lot and has been the reason for us to come to Puerto Rico. We started this whole endeavor that I'm going to talk about here in the presentation almost a year ago, just under a year ago. And so what I hope that you gather out of this is that we want to do more in space. We want to support more space-based research, and we want to do it at timelines that is very conducive to not just the industry, biotech industry, bioscience, biopharma industry, but also within the academic community as well. Looking at space as being accessible now, and the need to go to space being very broad, and it extends past what you typically would see as space-based research. And so I am going to go into, and it is talking about disrupting bioscience in space. I am with Rhodium Scientific. My name is Heath Mills. I am the Chief Scientific Officer for Rhodium Scientific. Our founder and CEO is Olivia Gomez-Holzhaus here. We are here in Puerto Rico again, I think now for the 5th time this past year. But what Rhodium is a company founded by Olivia in 2014. It was founded as a terrestrial company looking at ways of optimizing and making more efficient the process of discovery, how scientific labs move forward from the use of equipment to the overall standard operating procedures that labs do and use within their facilities. And it was seen then in about 2015, 2016 that the work that Olivia had started with the company could be used in space and to make products out of those missions, make the scientific process on orbit more like a terrestrial lab. And so that's where we moved in and transitioned in, and we are now one of the commercial service providers to the ISS National Lab. You may have heard Ray Lugo's talk this morning about the ISS National Lab. We are one of those companies that he was mentioning there. And our big forte and what we work on a lot is this translational process. How to translate what you do in your terrestrial lab to what is flight feasible, something that's able to go to space and develop new products, take advantage of the overall microgravity, low-dose radiation environment to produce something new and novel. And so it's that teaming effort that we work with you on to help you get to and realize that your work can and should be done in space. And so part of what, and I'll move out, sort of slide over, I guess. What we've done is, in a way to catalyze these opportunities to go to space, we've set up multiple challenge series. We've done one now for 3 years down in Australia. This was the first year that we've done the one here in Puerto Rico. Some of you all have been part of the challenge, and a couple other people are running around right now. They've also been part of the challenge. But what we did is we set up this challenge where we came in and started holding some workshops talking about how to go to space, why to go to space, that space was available. But what we wanted to do was take what is naturally the big benefits of doing research here in Puerto Rico, the industries, the biotech, the biopharma, build upon what has been very successful here in Puerto Rico and show that this can be going even further and advanced faster by going to the space environment. It's not a change of what's done, it's a way of doing what's now done well, doing it better. And that's what the challenge was about, finding a way to now open up those new markets by going to space. And yeah, sorry, I still have that picture in here. I'll stand right here. But we worked with the trust to develop these 2 different workshops, and it was the trust, it was Indoneve, it was 5G Zone, multiple players within the island to come together to develop 2 different workshops that happened back in April, where we highlighted what was it like to do an engineering project, what was it like to do new tech development, what was it like to do new bioscience in space, who should go and do a lot of tutorials to get and try and broaden the idea of I can also go to space. And so that became the question. Why would I now add space to my portfolio? Why would I now add space to what I do in the lab, in our facility, at our university? Why go and do this? And so, number one, it's there. It is a facility. It is a laboratory. It is available. It is the national lab. ISS National Lab, what Ray was talking about, is an organization. There is actually a true national lab within Destiny module here that is available. It is part of the U.S. infrastructure. It is there for us to use. Not everybody realizes that this is basically a field station, and it should be used and accessed as a field station to go and do research. Now, I am splitting up the rest of my time to talk a little bit more about that why. And this is what we brought in through the challenge. We had two different sides of the challenge. One of the tracks was for biotech and biopharma. Now, this is the science mission of the two tracks within the challenge. So you were able to choose which side you went into. For the biotech, biopharma, we asked the question of, why do biology in space? Why should my biology, my biotech, my bioscience go to space? Well, first off, it's a biological organism. It's a biological organism. It responds to its environment. There's a reason why a lot of people from Wisconsin come down to Puerto Rico in the wintertime, because they're responding to their environment and saying, I'm getting the heck out. It's much better here than it is in Wisconsin this time of year, any time of year. But biology responds to their environment. In space, there's two very novel stressors. There's two very novel pieces of the environment that cannot be reproduced back on Earth. And if you think it can be reproduced, come see me afterwards. It cannot be reproduced on Earth, and that is the microgravity environment and the low-dose radiation environment. It is two unique factors. And with life being evolved here on Earth over 4 billion years, those two factors are new for biology. Biology gets up and freaks out. The astronauts act differently, the bacteria act differently, plants act differently. So the whole point is to go up there and test what that difference really is. Now the difference, three main big characterizations of that difference can be seen in the genotype. Okay, that's the DNA. DNA changes, mutates. Some of those mutations can be good, some can be bad. Some can be good because it's doing what you want it to do. Some can be bad and it produces a new pathogen. It produces something maybe not so nice. But it is this gain of function mutations that are very interesting to go up and study. What can the organism evolve into up there that we haven't seen back here? A lot of good research in that area. The other one are the physiologies. Physiologies are more of the, I'm going to put on a jacket because I'm in Wisconsin. I'm going to get smart and come to Puerto Rico and I don't have to wear the jacket anymore. It's a temporary change. It's a shift in how the organism acts at that moment. So there are some organisms that get to space and produce a novel metabolite, a novel protein, a novel drug compound that you don't produce here on Earth. And so it's a temporary shift, and that builds into a little bit of the biomanufacturing that I'll talk about in a little bit. So this is temporary. Now this one is the functional advantages. The functional advantages are you don't have gravity. And if you don't have gravity, you can build a 3D structure that is much more fine. It's much finer scale than you can produce on Earth because you don't have deformation because of gravity. You don't have settling. You don't have the big particles falling to the bottom of the flask, the small particles staying up in the top. You can have a more homogenized mixture. If you're doing biologics or you're doing chemistry, you're doing polymers, this becomes real interesting. Not reproducible on Earth. So this is the why to go to space. Now, within those tracks, we then highlighted three main areas because from what we see and what the research that we've done really puts forward is there are three main verticals that within the biotech, bioscience areas that are showing good progress, good results. One of those is within regenerative medicine, and we were talking about Mark Vanderheyer a little bit earlier today, Mark helping us with one of our missions here. Within regenerative medicine side, astronauts on orbit experience some physiological traits, temporary traits, associated with diabetes, cardiovascular disease, different inflammatory immune diseases, neurodegenerative diseases. These things, these maladies that we see as an aging populations the astronauts start to get precursors, start to get signs of early onset of different age-related diseases. Most of them, when they come back to Earth, no longer have pre-diabetes, no longer have pre-cancer, no longer have those markers, which means the body regenerated itself, fixed the problem, which means we have the ability to recover from some of these maladies, but we lose them over time. If we identify what those triggers were that allowed the astronauts to recover, to revert, to regenerate, that could be interesting. And that's part of the regenerative medicine side. We've flown 2 missions right now. These are both with the gut microbiome. The gut microbiome is very important to overall health. Getting your gut microbiome out of sequence, dysbiosis is the big technical term that scores a lot of points in Scrabble. Looking at the way the gut microbiome shifts in orbit can start leading to maybe some causes that are seen here. So that was some of the projects that we went in and looked at. Moving that forward, and I mentioned it a little bit, that if you can identify what those triggers are that revert the body, or the triggers that cause some of those maladies to start taking shape, well, now you're talking drug discovery, drug development. Now you go after those metabolites, those production molecules, something that the body or the cell is producing that induces those states. If you can isolate those, you're going to go down to Amgen, you're going to go down to Merck, you're going to go to one of the facilities here on the island and start producing a new drug compound. Can it be produced back on Earth, or must it be produced on orbit? Well, if you produce it on orbit, now you're talking about biomanufacturing. And this is our third area that we're looking at. And this is what a number of the proposals within the challenge targeted, was biomanufacturing. Biomanufacturing is using biological organisms to produce a molecule, something of interest. Maybe it naturally produces it, or maybe you have to do what is called, the new novel term is synthetic biology, because genetically modified organisms is a bad word these days. So you now say synthetic biology, and everybody goes, yay, same thing. So we'll call it whatever it is to get the funding and go do the project. But same thing. But bioproduction, biomanufacturing is now, you've identified a compound of interest, let's go produce it. So that's one side of it. The other one is, and what a lot of organizations, and especially you start seeing some Department of Defense logos down here, you're also very interested in point source manufacturing. When you're working in an extreme environment, an environment that has resource limitations, that may have a supply chain that may be very difficult to get to. It may be like you're on an island. Biomanufacturing may be very important. And so now using the biology to produce those compounds. This is something that we at Rhodium have had a lot of success with. We've flown a lot of missions on. We have been able to really go out and produce some interesting results using biomanufacturing within the space-based environment because of those novel physiologies on orbit. They change their growth rates. If you change your growth rates and we want you to grow faster to produce something, maybe you can produce a better and more efficient product on orbit using the LEO, the low Earth orbit environment, to do it. And so that's one of the big things that we looked at. Now, the other track, that was the biological side. And we went through and we have the workshops. They're actually online. If you go to the trust, they're still online. You still get to see my head bouncing around on those talks. That was the biological track. And we got a lot of university and some industry partners coming in and applying to the challenge for that track. The other track was in tech development. There are a number of reasons to do tech development in space. One, you're looking at not everything that is built for Earth can operate in space. One of the little tests, the easy, simple test is, can you turn over whatever you've built, whatever you've designed, can you flip it upside down and does it still work? Not everything, if you flip it upside down, still works. A beaker does not work upside down. So if you flip it over, you have a chance. Outside of that, ruggedizing this for the radiation environment, for the launch sequence, for the landing sequence, for the astronauts kicking off of your piece of hardware that you've designed, becomes a challenge. Miniaturization. Space is infinite. There's not infinite space on the space station. So now you have to take something that has a nice big footprint on your bench top and make it smaller. Can you miniaturize it? If you can miniaturize it, you can send it to space. If you miniaturize it, you can put it on a vehicle. If you miniaturize it, you can send it to sea. If you miniaturize it, you can send it to a forward operating environment. There's a lot of industry out there for something that's miniaturized and ruggedized, not just space. But space is a great place to go. And then overall, it's a test environment as well. Can you operate in a high radiation environment? Can you operate in a vacuum? Can you operate with the extreme environments, temperature swings that you see in space? Let's go test it. So all of these became some of the reasons to go do tech development on orbit. We have gone in, and at Rhodium, we've developed some of our own equipment, some of our own hardware, some of our own flight-proven and tested hardware, using everyday lab equipment. Sometimes you don't have to reinvent the wheel. Sometimes you use what is there, what the scientists understand and have developed a very broad data set for, and you fly it. One of our missions, biomanufacturing missions, we used small vessels, small tubes that our ground teams tested over 1,000 of them before we went to orbit. We can do that because of the way we design the equipment. You can optimize your hardware. But now what we were looking for was the what's next. What is out there? There's a lot of very good engineering on the island, a lot of very good med tech development done on the west side of the island, looking at ways of finding that what next. And that's what the challenge was then used for, was to go in and do that. And so that is what we then did. So over this past year, we developed this with the Trust, with Indoneve, with the different partners, with 5G Zone, and developed this whole process to stimulate many proposals to go forward. Now, with any challenge, with any environment, with some things other than the whole participation award, but that's silly, is that there's going to be one winner. But what we did with this challenge and what we've done with these teams is that we've worked with the teams to develop. All of these are now flight feasible. All of these should go to space. And so what we're doing now at Rhodium is after the winners are going to be announced in January, we're going to start taking some of those now proposals and let's fly them all. Let's find other homes for them. Let's move them forward. So as we roll into year 2 next year, and you'll see hopefully more announcements like this coming up in April, June, July for round 2 of these, remember, you get consulting on this, you get our efforts on this. In the end, you're going to have a proposal that is flight feasible that can be taken to NSF, NIH, pick a DOD, pick a set of letters, pick an investor, pick something, because this is a mission that can fly and should fly. And that's what we did with the challenge. That was our big overall goal with it. And if you want to get started on one of these missions, whether it's with the challenge series or outside of, the QR code is active and does work. And you'll see that some around the building now that we got some printer back from Office Max. Another fun story. Scan the code, and that will go straight to us and straight to our team, and we will start working and developing your projects. So with that, and 36 seconds left of a very late presentation, thank you all very much, and open up to any questions. You can't ask a question. Yeah, please. Yeah.
Some of the changes that you observe, physiological changes, they were observed as a result of the space, those regenerative changes were observed as a result of the mission?
They were as part of the mission. So that becomes the then challenge, is what was the overall driver, the biological driver of that physiological change that you identified? What was the stressor that did it? Was it the radiation? Was it microgravity? Was it the launch sequence? Was it something involved? What we were trying to do with our cell cultures, with our microbiological experiment, being a microbiologist, I should be able to say that word better, is to eliminate the flask effect. We don't want the effect to be the hardware that the biology is growing in. And that's where the optimization side comes in. So we then go through and with the right controls, with the right overall aspect of the mission, we eliminate the launch and landing effects. We eliminate those different variables to really constrain the experiment to be, no, this was because of 6 days of growth on orbit, or 20 days, or whatever it is. And so that's what we're trying to do on that. Also, because of the size of the vessels that we're using and the numbers of vessels, both of our last 2 biomanufacturing missions had 216 samples. And so we're trying to get the replicates, these actually quadruplicates on these, to get the statistical side on this so we can start saying that. Unfortunately, yes, we've been going to space 60, 70 years now. Not all experiments done on orbit have been done to the caliber that you would do them in your lab here. And because of that, your data sets tend to show trends. Squint sideways, put your head on it, and as you can clearly see, the data doesn't always fall. So that's what we're trying to get out of right now and find out what those isolated variables are that's causing that physiological shift. Yep.
And you're finding correlations?
Yes. Finding very unique ones. We're publishing a paper right now on one of the projects that, I don't know if it was up here or not. I'll go back to it real quick. I promise I'll stop. I appreciate that. Here, this guy here, phage evolution, where the publication is in press right now for this one. The researcher on this, lead researcher on this from the University of Wisconsin, Vatsan Raman, long-time study of phages. Phages are viruses that only attack bacteria, can be used as novel therapeutics, moving away from antibiotics, that whole side. Looked at the mutational structure of the phage when it returned. The mutations that we saw, over 20,000 unique mutations within the phage that we saw on that, were not and have never been seen on Earth. Now the challenge is to take the mutational analysis that we've seen on this and then attribute it to either microgravity, radiation, most likely radiation and microgravity, but also the physical environment that microgravity produces, no convection. So it allows for a different landing structure to be evolved. That's a unique space environment. How can we then back engineer that to a new therapeutic that makes penicillin look like nothing? That would be fun. But it's little things like this, and because we're doing the data sets and the post-flight analytics to that degree, we're now going in and starting analyzing. And that's the fun part for us. Yes, we are the implementation partner. Yes, we provide space hardware. Yes, we go to launch, but we're also the scientists on the team. And so we're co-PIs on all these projects. And so that's my fun side. I get to go back and wear my genetics hat because I'm a molecular genetics guy. So that's where we get to have some fun with it. Yeah, that's right, true. Not on this one. Next slide. Synthetic cryptobiology, this one here. This one's also is now in print, or in press. Looking at protection of DNA, moving DNA from point A to point B. So you encrypt either a function or information on DNA, a plasmid at this point, and then move it from lab out at Berkeley through the space station back to Berkeley, and we preserved 100% of the DNA code. Now that works real well with biomanufacturing because now if you have a novel product you're wanting to produce, but you're not wanting to produce it now, you're wanting to produce it on the moon, like these guys are doing, you can now ship it to the moon, and when you're ready for it, you resuscitate the organism, and now you can have that function. And so we tested the capability of moving, and that's why it's synthetic cryptobiology. We were using biology to encrypt. It wasn't crypto coins, which have been a lot more, yeah. But it was maintaining the DNA structure and the information, and it worked. We got 100% sequence recovery after going to orbit and coming back without any cryopreservation, without any special treatment. This was one of those that we told the astronaut just to beat the mess out of it. We don't care. We want to see what happens to it when it comes back. Don't treat it nice. Can we get it back? Have we done our job biologically to preserve it? And now it can be used anywhere. So it worked. Thank you. Good. Yeah, Matthias.
Follow it on that question. If you have to protect your DNA,
Mm-hmm.
what protection do you put to your microorganisms that you send out there?
So depends on the final outcome and what we're trying to do with it. So yes, the chassis organism, and that's what the Be Sure program here is all about. Biological surety, beyond Earth. It's an acronym, man. I work with NASA. There's acronyms embedded in acronyms. I'll come up with that Be Sure was. But this is about biomanufacturing. And with the biomanufacturing, protecting of the organism is as obviously important as the information the organism has. What this project, so we kicked off this mission in November of last year. We flew it in March of this past year. This one was then designed based upon the success of the first mission, and we flew it in August. So we've had 2 missions with DARPA within the last year, and the third one's going up in March. The whole point there is protecting and developing the chassis. The chassis is the organism that then produces that biological product of interest. And so it's ways of producing a better, more hardened biological structure to withstand the rigors of the space flight, the requirements of space flight, the frozen, the thaw, the long-term stay, the radiation, and all of that. Can we develop a product here? And the goals that the DoD put forward on this mission was go first, produce 25% more product than you can produce on Earth. We did that. So they said, okay, now do 50. We did that. And so now we're moving it forward by using some of our optimization products that we're doing to protect the organism. So also very good.